Hi everyone, my name is Sarah and today I'm going to be discussing randomization and its importance in clinical trials. So hopefully at this point in the semester you all have a good understanding of randomization and the different techniques used to achieve it. However, I know personally I can always use a quick refresher on the more basic topics. So I hope this presentation can be an easy listen for you and will maybe provide you with a piece of information you might have forgotten throughout the semester. So I would first like to just give you guys a breakdown of what we're going to be talking about in today's presentation. Before getting into the different types of randomization, I'm going to explain what it is and why it's important. So randomization is a sampling method that is often used in research and clinical trials to assign participants or subjects to groups by chance as opposed to by choice. So the main goal of randomization is to prevent bias as much as possible and to produce groups that are equal and therefore comparable. As we've just seen, randomization prevents selection bias and produces comparable groups. So it is really a method of experimental control in a study. By ensuring that participant groups are equal, you will be confident that any changes that come up in the results are due to the treatment and not due to some other variables. Randomization also ensures that physicians or health professionals are not choosing which patients are going to receive a potentially beneficial treatment over the other person. Increasingly, evidence-based practice is placing an importance on rigorous and well-conducted clinical trials, and randomization just happens to be one component of this. To be more specific, in clinical research, randomized controlled trials are considered the gold standard for determining effectiveness of a treatment. So it's important to understand the different types of randomization and how we might achieve it in a clinical trial. So before I flip over to the next slide, is there anybody who might be able to provide me with one or two examples of the types of randomization? Good. So although there are many different types of randomization, we're only going to cover a couple here today, but as you can see up on the slides, there are several different methods. So to start off, I'm going to explain a very basic method known as simple randomization, which is based on a single sequence of random assignments, and it's most often achieved by flipping a coin. It can also be achieved by using a deck of cards, rolling dice, or using a set of computer-generated numbers. This method is great because it is very straightforward and very easy to implement in clinical trials. However, some problems can arise if it's being used in small populations, since it can result in unequal group sizes. What's more, this method does not tend to work well when the population is very diverse. In contrast to simple randomization, where each possible sample is equally as likely to occur, stratified sampling methods divide the study population into groups based on certain characteristics or covariates. So researchers must first identify these characteristics and determine their potential influence on the results, and then they can create different blocks based on this. Then they can separate the participants into these blocks, and then simple randomization is performed within each block to determine who is going to receive which treatment. This method is also quite simple and easy to implement, and it's sometimes chosen over simple randomization as it ensures that each subgroup is equally represented in the sample. And although this might seem like a better option, it still has its disadvantages as well. One example being that as the number of characteristics increases, it becomes increasingly complex to control for them. So circling back to what we've already learned, randomization is the process in clinical trials to sample patients and assign them to groups based on chance and not by choice. Randomized controlled trials are considered the gold standard for determining efficacy of a drug, since the randomization strengthens the results of the study by eliminating selection bias and producing comparable groups. And although there are several different methods to achieve randomization, we've only covered a couple here today, so I strongly encourage you to go back and review the different types and their respective advantages and limitations. And that brings me to the end of my presentation. So I hope you've all maybe been reminded of something here today and maybe take it as an opportunity to go back and review what we already know from the semester. Thanks, guys.